Ready to unlock some serious secrets. Today, we're diving deep into the world of genetics. We're talking DNA, RNA, and even those Punnett squares you might remember from back in school. Ever wonder how we can trace things like eye color or height back to these tiny molecules?
It really is amazing when you think about it, right? How these elegant little mechanisms are translating genetic information into, well, life itself.
Okay, so let's start with the basics. DNA. We hear it everywhere, but what is it really? Like, what are we even talking about?
Imagine a library, right? A huge library filled with instruction manuals for, well, you. Each cell in your body contains a full set of these manuals, and that's your DNA. This instruction manual, all packaged up neatly into chromosomes, determines everything about you. And you know what's really wild? All that information. It's encoded with just 4 letters: A, T, C, and G.
It's like a 4-letter alphabet that spells out the instructions for an entire human being. Crazy. But, I mean, it's not like we can actually read those A's, T's, C's, and G's, right? How does DNA actually work?
You're right. We need a way to decode those instructions. That's where RNA comes in. Think of DNA as like the master blueprint kept safe in the cell's nucleus. RNA, that's the working copy. And there are actually different types of RNA, each with its own job to do. But they all work together to translate that DNA code into action.
So DNA is kind of shouting instructions across the cell, and RNA is like the messenger running back and forth.
Exactly. One type of RNA, it's called messenger RNA, or mRNA, and this carries a copy of those instructions from the nucleus out to the protein-making factories of the cell. Those are called ribosomes.
Okay, so we've got the instructions out in the open. But how do we go from instructions to actually assembling, let's say, baking a cake? How do those ribosomes fit into all of this?
Ribosomes, think of them like incredible little machines, molecular machines that can actually read that mRNA, that recipe card, if you will, and then link together the building blocks of proteins. Those building blocks are amino acids, and the ribosomes link them in the right order.
Hold on, hold on. How does the ribosome know which amino acid to use at each step? It's like having a recipe that just says, add a pinch of this and a dash of that. How does it get translated so precisely?
This is where things get really mind-blowing. That sequence of A's, T's, C's, and G's on the DNA, that determines the sequence of building blocks in the final protein. Every 3-letter word on that mRNA recipe card, we call that a codon, corresponds to a specific amino acid. It's like a secret code, and the ribosome has the key to decipher it.
Wow, so there's a whole language within our cells. DNA's 4-letter code gets translated to the 20-letter language of proteins. So we've got DNA as the blueprint, RNA is the messenger and the builder, and ribosomes are the chefs whipping up proteins according to the genetic recipe. This is amazing. But how do we even figure all this out? This seems like science fiction.
Well, that's where Gregor Mendel comes in. Remember him? He was a monk back in the 1800s who had a real knack for gardening and a very curious mind. And just by observing how traits were passed down in pea plants, he stumbled upon some really fundamental principles of heredity.
You mean like those Punnett squares and dominant and recessive genes? All that stuff we learned in school? That was Mendel.
Exactly. Mendel's work, all those meticulous notes on his pea plants, that laid the groundwork for everything we understand about how traits go from one generation to the next. And get this, he didn't even know about DNA or genes yet. But his observations, those set the stage for a revolution in biology.
Okay, so Mendel's pea plants were kind of a big deal. Tell me more. What did he actually discover?
Well, Mendel, he figured out that traits are determined by these distinct units of inheritance. And today, we call those genes. He saw that offspring, they inherit 2 copies of each gene, one from each parent. And sometimes, just one version of a gene can be dominant over the other, meaning that trait will win out even if there's only one copy present.
So that's why some traits seem to skip a generation. It's like a genetic coin toss, and sometimes the dominant side wins, sometimes the recessive one gets its moment. But how does all of this connect back to DNA and protein synthesis? So we've got Mendel's laws, right? They explain how those traits get passed down. Then we've got this whole intricate dance of DNA and RNA and proteins happening at the molecular level. But how do those two things even connect? It's a pretty big leap from looking at pea plants to, like, understanding the language of our cells.
You're right, it was a huge leap. It took so many brilliant minds and decades, really, to make that connection. But basically, Mendel's factors, those units of inheritance, they're actually segments of DNA. And those segments, they code for specific proteins.
Okay, so when we're talking about a gene for, say, eye color, what we're really talking about is a stretch of DNA. And that DNA holds the instructions for making a protein. And that protein helps produce the pigment in your eyes.
That's it. And those different versions of a gene that Mendel was observing, the dominant and recessive alleles, those are really just slight variations in the DNA sequence, small changes that can lead to, well, big changes in the protein. And that's how they ultimately affect the trait.
So a single letter change in the entire DNA code could be the difference between blue eyes and brown eyes, all because it affects how a protein is built, how it does its job.
Exactly. That's the power of genetics. Even tiny changes happening way down at the molecular level, they can have huge effects on our traits. And, the more we learn about all these connections, the more we realize just how incredibly complex and interconnected the world of genetics really is.
It's like this massive puzzle, right? And each piece, each gene, each protein, it all fits together to create this bigger picture. It's incredible.
Well,
But hold on, we haven't even touched on the really futuristic stuff yet. Gene editing, DNA fingerprinting, gene therapy. I mean, it seems like every day there's some new headline about how scientists are using genetics.
it really is a remarkable time to be studying genetics. We've gone from, well, observing patterns in pea plants to mapping out the entire human genome.
And not just mapping it out, but changing it. Gene editing tools like CRISPR, those have totally revolutionized things, right? Now we can actually make these really precise changes to DNA sequences. It's like having a scalpel, but for genes.
It's true. The applications are astounding. We can modify crops so they resist disease or drought. We can develop therapies for all kinds of genetic disorders. We may even be able to correct some genetic defects, in embryos.
That's incredible. But also, like, a little bit, I don't know, intimidating. With great power comes great responsibility, right?
Ditto.
Seems like every breakthrough with gene editing comes with a whole host of ethical questions.
Absolutely. The ability to actually alter the very building blocks of life, that definitely raises some profound questions. As a society, we have to grapple with these. We have to talk about the potential benefits, but also the risks. And we need to figure out some clear ethical guidelines for how to use these technologies.
It's a lot to unpack. But we can't forget about things like DNA fingerprinting. It's not just something you see on crime shows now, is it?
Definitely not. DNA fingerprinting is being used everywhere. Forensics, ancestry research, it's become a really powerful tool.
So it really is like a genetic barcode. We can actually use it to identify people with incredible accuracy. No more cases of mistaken identity.
Exactly. But it's not just about identifying individuals. It can also help us track how diseases spread, look at migration patterns, even trace the evolutionary relationships between different species.
It seems like the possibilities are endless. And speaking of pushing boundaries, what about gene therapy? I mean, using genes themselves as medicine, that just seems mind-blowing.
Gene therapy, it has immense potential for treating so many different diseases. And we're talking everything from inherited genetic disorders to certain types of cancer. And the idea is actually pretty simple, at least at its core. We're delivering functional copies of genes into cells that either have a defective copy or are missing that gene entirely.
Oh, okay. So it's kind of like taking a faulty part in a machine and replacing it with a brand new one. Except we're talking about fixing errors in our very DNA.
Precisely. And while it's still early days for gene therapy, we're already seeing remarkable success. There are certain types of inherited blindness, for example, that are being successfully treated right now with gene therapy.
It's pretty amazing to think that we might actually see a future where these genetic diseases are a thing of the past. But, like you said, it's still early days for gene therapy. What are some of the challenges that researchers are facing right now?
Well, one of the biggest hurdles is figuring out how to actually deliver these therapeutic genes and how to get them into the right cells. We can't just, inject a gene into the bloodstream and hope for the best. It doesn't quite work that way.
Right, it's like sending a package without an address. So how do they make sure those genes get where they need to go?
Well, scientists are coming up with all sorts of incredibly ingenious delivery systems. Often they're using viruses, but like disarmed viruses, as vehicles to deliver the therapeutic genes.
Wait, viruses?
Yeah.
Delivering something good? That's kind of ironic, isn't it?
Mhm.
I mean, they're usually the bad guys.
It's true, but if you can remove the harmful parts and actually engineer them to carry these therapeutic genes, well, you can take a former foe and turn it into a powerful ally. It's pretty remarkable.
That's wild. But I have to imagine there's still so much that we don't know, especially about the long-term effects of things like gene editing and gene therapy. It feels like we're really stepping into uncharted territory here.
You're not wrong. It's a brand new world, and we have to be very cautious. We have to constantly be considering the ethical implications. These technologies have incredible potential, but at the end of the day, we have to remember that genes don't exist in a vacuum. They interact with each other, with the environment, in ways that, frankly, we're only just beginning to grasp.
It's really humbling when you think about it. For all our scientific advances, we're still really just scratching the surface, aren't we?
Absolutely. But isn't it exciting? The more we learn about genetics, the more we realize just how much more there is to discover. It's a field that just keeps challenging our assumptions, pushing the boundaries of what we thought was even possible.
This deep dive has definitely given us a lot to think about, that's for sure.
Yeah.
From those elegant double helixes of DNA to, well, these really profound questions about what it means to manipulate the very code of life. I think if I've learned anything, it's that the world of genetics is full of surprises,
Yeah.
and it's bursting with possibilities.
And maybe the most important takeaway here is that genetics, it's not just about genes, it's about understanding life itself, that incredible, intricate tapestry. And it's about using our knowledge wisely to hopefully create a healthier and, yes, a more equitable future for everyone.
I think it's a great place to leave it. So as we wrap up this deep dive into the fascinating world of DNA, RNA, and Mendelian genetics, we'll leave you with this thought. What will your genetic legacy be? How will you use this knowledge? It's a lot to ponder.